Alright, so today we're going to be talking about metabolism and DNA synthesis.
Hi, today I will walk through how ATP is made, starting from glucose and carbon dioxide, and how this ATP is used to make nucleotides for DNA replication. I will follow the metabolic pathways shown on my poster, including glycolysis, the citric acid cycle, the pentose phosphate pathway, nucleotide synthesis, and ATP production through ATP synthase. Starting materials, glucose and carbon dioxide. So looking at the poster area with glycolysis plus PPP start, we begin with glucose as the carbon source and CO2 as an end product of metabolism. Glucose enters glycolysis where it is broken down to pyruvate, producing ATP and NADH. Some glucose is also derivated into the pentose phosphate pathway to make ribose for nucleotides. Glycolysis pyruvate to acetyl-CoA. In glycolysis, glucose is converted to pyruvate. This produces a small amount of ATP and NADH. Pyruvate then enters the mitochondria and is converted into acetyl-CoA, releasing CO2. And then acetyl-CoA enters the citric acid cycle. Citric acid cycle into NADH, FADH2, and carbon dioxide. In the citric acid cycle, acetyl-CoA is oxidized to CO2. This produces NADH and FADH2, which carry high energy electrons. These electrons will later be used in an electron transport chain to make ATP as energy. Pentose pathway into ribose 5-phosphate. Some glucose 6-phosphate enters the pentose pathway. This pathway produces ribose 5-phosphate, which is used to make nucleotides. Ribose 5-phosphate is converted into PRPP, which is then activated sugar used in nucleotide synthesis. Next, we have nucleotide synthesis into AMP, GMP, and ATP. PRP is used to build purine and pyrimidine nucleotides. These are converted into AMP, GMP, and other nucleotides through phosphorylation. ADP becomes ATP. ATP is required for many cellular processes, including DNA replication. Electron transport chain. NADH and FADH, in looking at the ETC membrane drawing, donate electrons to the electron transport chain in the mitochondria. As electrons move through the chain, protons are pumped across the membrane. This creates a proton gradient that stores energy. ATP synthase and ATP production. Looking at the ATP synthase drawing, the proton gradient drives ATP synthase, and from this protons flow through ATP synthase, causing it to rotate. This energy is used to convert ADP and phosphate into ATP, and this is the main source of ATP in the cell. ATP used for DNA replication. So we're going to be looking at the nucleotide, DNA, and PRPP area. ATP is used to make deoxynucleotides needed for DNA replication. DNA polymerase uses DATP, DGTP, DCTP, and DTTP to build DNA. This energy in these nucleotides drives formation of phosphodiester bonds, which then allows DNA replication to occur in the cell. Starting from glucose and carbon dioxide, the cell produces ribose, nucleotides, and ATP. ATP is generated mainly by ATP synthase using the electron transport chain, and this ATP is required to make nucleotides and to power DNA replication. And finally, altogether, these pathways all have their own working gears and work together to connect metabolism to DNA synthase. And without one of these steps working, the whole cycle and the whole space of this will not work. Thank you so much.